Hello, I'm Jesse McQuarters, Brand and Content Editor for STAT, and I'm thrilled to welcome you to this STAT Brand Studio virtual event. STAT's gatherings, both in person and online, bring together communities of thought leaders and trusted industry experts across the life sciences, medicine, and healthcare. With these sponsored partner forums, we're joining with visionary leaders to discuss the trends and topics that are shaping the world. Thanks again to all of you for being here. And of course, I'd like to remind everyone to become a STAT Plus subscriber and check out statnews.com for the latest coverage in health, medicine, and the life sciences. It gives me great pleasure to welcome to the STAT Brand Studio stage two guests working at the forefront of biologics manufacturing. Doyun Park is an Associate Director of Corporate Engineering at Samsung Biologics. He has over 20 years of combined experience in managing cGMP small molecule HPAPI manufacturing facilities and CapEx projects. Thomas Rohrer is a Senior Director of Bioconjugate Technology Support at Samsung Biologics. He has an extensive background in the biologics industry with more than 38 years of experience in biological process development, clinical manufacturing, and licensed biological manufacturing. First of all, thank you both so much for joining me here today.
Yep. Thank you, Jesse. It's a pleasure to join you.
So I'm going to kick things off and just pass the baton. But I know you want to start off by looking at ADCs past, present, and future. And I'll just, again, pass the baton over to you and let you give some insight into some of the historical context of ADCs, at where we are now, and the future going forward.
So we're going to cover various topics in today's presentation. We'll start with a brief introduction to ADCs. We'll talk about antibody-drug conjugates themselves. What are these unique creatures? We're going to talk about facility design and support of the technology itself, and safety considerations for ADCs in manufacturing, because you are dealing with highly toxic drug payloads, which are being conjugated to antibodies. It's necessary to delve into the safety considerations for personnel working in those particular areas manufacturing ADCs. And then we'll have a discussion at the end. So next slide, please. Next. The history of therapeutic ADCs dates to 2000, when Mylotarg was approved for treatment of acute myeloid leukemia. A decade later, with advances in linker and payload design, FDA granted accelerated approval to Seattle Genetics ADCetrus. In the case of ADCetrus, the cytotoxic payload consisted of a potent anti-mitotic agent, monomethyl auristatin E, and it was the first new therapy approved since 1977 for the treatment of Hodgkin's lymphoma, and was the first therapy to receive approval for the treatment of anaplastic large cell lymphoma. Following on the heels of ADCetrus, Roche Genentech received approval for Kadcyla for patients with HER2-positive metastatic breast cancer. And currently there are 15 ADCs approved for antibody-drug conjugates. And it is estimated there are 300 ADCs in various stages of clinical development. Notably, a few novel ADCs have established a substantial anti-tumor activity in many tumor types and that share expression with the target antigen. For instance, HER2 overexpressing breast, gastric, and colorectal cancer and HER2 mutated lung cancers have demonstrated a significant response to Enhertu. Recent approval of ADCs for difficult to treat therapeutic indications because of improvements in the technology and also better understanding of tumor biology have led to renewed interest in ADCs. Next slide. the future prospects for ADCs, we see a significant expansion in the requirement for ADCs. In fact, in moving from this year out into 2029, we're projecting over 2.5x increase in the need for capacity for manufacturing ADCs. Of course, manufacturing these unique drugs require a very in-depth understanding of how they work in their complexity, because you have three agents, both a targeting agent, a linker, and a payload that can be tuned for their therapeutic indication. We also see diverse technologies being developed with respect to site-specific conjugation to reduce isoforms in ADCs. And Samsung as a company is certainly exploring these technology trends and seeks to engage with companies that are leading stakeholders that are really spearheading the innovation in the field. Next slide. We'll talk a little bit about an antibody-drug conjugate. So the components of an ADC really consist of the antibody, the targeting agent, the linker, and the payload. The targeting agent really enables precise location and binding to a specific cell population. The linker is a bifunctional chemical entity that has the ability to not only link to the antibody but also its payload. And moreover, the linker can be controlled in such a way, depending on its design, to release the payload in specific locations, depending on the therapeutic indication. The payloads typically are cytotoxic drugs that kill cancer cells through various mechanisms such as inhibiting cell growth or inducing cell death. In this particular slide, I show an SMCC linker, which is a non-cleavable linker attaching DM1, which is a myotansine, and it is attached to the antibody by a free amine. But suffice it to say, there are many different configurations of ADCs, but this gives you a general overview of how these unique drugs look. Next slide, please. So, we'll talk about facility design and the supporting technologies. Next. So the general manufacturing process in facility design certainly needs to take into consideration safe handling of cytotoxic compounds, with employee safety being of the utmost importance. And of course, this all has to occur in an aseptic biopharmaceutical environment. And this environment has to provide prevention of cross-contamination. Usually the suites that are designed are dedicated for ADC, and they have to be capable of, in the case of a CDMO, multi-purpose in the case, such that various campaigns of ADCs can run through the facility. Next slide. So if we look at a general process overview of an ADC conjugation and how it operates, it usually starts with modification of the antibody in some way. That could be, in most cases, reduction of the antibody, or maybe it's activation of primary amines. In almost every case, it means that the storage buffer for the antibody is converted over to a buffer system that would be amenable for conjugation. Next, the process parameter. No, if you can back up, please. Now you're moving forward. Back. Okay. So scale up of the equipment is also critical because the majority of these experiments are run at 2 mil to 5 mil scale because of the expense of the raw materials. So having a good model system in terms of mixing and control of the reaction stoichiometry is critical, and being able to scale it from very small scale up to your maximum manufacturing scale of 500 liters in our case is critical. And then next, the design of the equipment and cleaning procedures for multi-product facility application. Next slide. So if we look at the general process flow, we usually start with the bulk MAB, which would be run in a MAB, what we refer to as MAB modification suite. So it would be a 4-suite arrangement that would consist of MAB modification, the isolator suite, where the toxin and payload are brought into a solvent to solubilize it. Then we move on to conjugation suite, and in that conjugation suite we combine the frozen, maybe modified MAB with the cytotoxin under the conditions that are required for the reaction. And then following the reaction, you will have free payload that is more than likely left over from the conjugation reaction that needs to be removed. And typically UF ultrafiltration, diafiltration is the means for removal of free payload and residual solvent. But in some cases chromatography is required if further segregation of the isoforms is required. And then from that you move directly to formulation, which is in a separate formulation suite in which excipients are added to the drug substance to stabilize it before freezing or storage before it goes for fill finish. Next slide. So I'll mention a little bit about the analytical support that is required for ADCs because we have facilities and utilities that are supportive of biological, aseptic biological manufacturing. Many of the environmental monitoring, water sampling tests, and so on are the same you would find in any aseptic biological manufacturing facility. But when one comes to raw materials, in process control testing, biochemistry, and so on, that's where we start to see a number of analytical methods that are specific for ADCs. In the case of this slide, those that are dark in color are methods which are typically used for the manufacturing of antibodies, naked antibodies, and those in blue are specialized methods that we've had to add to be supportive of antibody-drug conjugate drug substance manufacturing. Probably one of the most complicated of these is a bioassay, cell-killing based assay that we've had to establish that will be part of a potency assay for release of drug substance. And also such assays as residual solvent that we use GC for to determine to assure ourselves that we have no residual solvent remaining from the conjugation process in the drug substance. And of course the all-important DAR value, which is the drug to antibody ratio, which is quite important and largely dependent on the method used. For example, HIC is quite often used for cysteine-based conjugations, but we very well may be using a different technique for lysine-based conjugations. Next slide. So with that, I will turn it over to my colleague, Darren Park. Darren is going to discuss the safety considerations for ADC manufacturing. Darren?
All right, thank you, Thomas. Yeah, so I'll be talking about safety aspects for ADC manufacturing and some of the thoughts that went into safety design as we were building new ADC facility. Next. Yeah, so we recognize that the managing safety was one of the most critical aspects of the business. So as an organization, we have the responsibility for the safety of the workers, but also as a CDMO, we wanted to ensure that we meet the client's expectations and their safety standards. So we really focused on two things as we were building the ADC facility and, thinking how to make this facility safe. So two aspects, right? The first one was on the facility design aspects. This is really referring to the physical elements of the facility, such as containment systems, clean room designs, pressure controls, and HVAC and air exchange and so on. And these are related for containment of high potent compounds to keep the place safe. Then the second aspect of the ADC manufacturing is really focusing on the training and management aspects, right? I mean, you build a system, but then you have to really efficiently manage it in order for the system to work. And so, some of the focus areas were, having the ability to run the potent compound handling program with the right talents, with the clearly defined roles and responsibilities across all departments. And then we really focus on having a good safety training, and then as an organization, have a capability to address safety-related issues. So these two elements really go hand in hand, and they enable a successful ADC manufacturing. So then the next, I want to talk about a little bit of, engineering controls. And then, how do you really know that you have the right engineering controls for ADC, right? I mean, the question is, you have the isolator, but then do you really know that it's the right one for you? And then this type of evaluation starts with risk assessment. And there are two parts to the risk assessment. The first part has to do with the hazard assessment. And this is really about understanding the physiological and toxicological properties of ADC and payload. Essentially, the question is, how hazard is the ADC? And then the outcome of the hazard assessment is OEL and OEB. And these are essentially used to communicate potency of potent compounds. Then the second part of the risk assessment is exposure potential. And this is really analyzing operational steps and identifying which steps contribute to exposure of potent compounds to workers. So, those activities that contribute to significant exposures are like labor-intensive steps, such as weighing and dispensing, high-energy steps, such as pressurizing the lines, and the physical forms of compounds. So, for instance, let's say you're weighing a payload and it's a dry powder, and then as you scoop some out, you can imagine that it will be dusting in the air. So immediately you will think that this will pose a danger to the workers without any PPE. So this type of exposure potential is called qualitative assessment. And then the exposure potential can also be identified by direct measurement too. So let's say there's a way to measure the concentration of payload in the air while performing the weighing and dispensing, right? Then you know the exact concentration. And this is called quantitative analysis. In either case, through the risk assessment, evaluating both hazard and exposure potential, you'll get a risk scoring. And based on the risk scoring, we can determine the type of containment. Before we move on to the next slide, we can talk a little bit about the OEL and OEL without going too technical. Essentially, OEB is called occupational exposure bands, and it's a categorization of hazard. So imagine like a hurricane, right? When you see a hurricane, it's very destructive, but then you rank hurricane based on category. So let's say Hurricane 4, you know that it's more destructive than Hurricane 1 or 2. So OEB is the same thing. So when we talk about OEB material of category 4, you can imagine that it's a lot more potent than something that's OEB 1 or 2. OEL, on the other hand, is a number. It's a concentration of airborne potent compound, which is likely not cause any health issues to workers for an extended period of time. And normally this is used to select isolator system or containment system. And for OEL, the lower the number, the more potent it is. In order to use the OEL properly, you need to have a good analytical method, because the concentration of OEL it can be quite low. So you really need to have a very sensitive method in order to analyze it. Normally, OEL and OEB are developed by or provided by the drug developer, but SBL has the capability to develop it for the clients and also develop the method too. So we talked about at Samsung Biologics how we use the risk assessment approach for designing safety elements in the ADC facility. So we were just following a very classical industrial hygiene process methodology as shown here. You know, the 4 principles of industrial hygiene process is shown, which is the anticipation and recognition of hazards, evaluation of exposure risk through qualitative and quantitative analysis, and selecting controls, strategies to mitigate the risk. So at Samsung Biologics, you know, we have developed something called control matrix for the labs and for manufacturing. And then essentially these are the guidelines for selecting the right containment systems. So, and then the last thing that I want to point out here is that there's a concept of confirming whether the controls are adequate. And this is done by taking samples of the air and surface while you're performing the work and compare those samples to the known OEL. And then you want to really make sure that those samples are actually stay under the acceptable level compared to the OEL. And then you have to continue to do that throughout the life cycle of the facility. Next, I want to actually kind of share an example of manufacturing control matrix that we have developed. So let's assume that we're handling payload category 4 material, and you're dissolving solvent and transferring the dissolved payload into a conjugation vessel. So based on the control matrix, open handling is not permissible during powder handling, and it has to be done in the isolator. Also, the transfer requires direct connection and closed transfer system with ventilation. So again, you know, there's no open transfer. Now, this is an example of a primary containment control matrix. And, you know, as an organization, you have to develop not only for the primary containment, but also for the secondary containment. So the example of the primary containment has to do with keeping the hazard material at the source of generation. But in case something happens with the primary containment, the secondary containment is about minimizing the spread of the material that escaped the primary containment. So, you know, for the secondary containment, we're talking about the HVAC, room differential designs, airlock designs, and material and personnel flows. So far we talked about the facility design, but, you know, it's really important to talk about the training and management aspects of the ADC facility, right? I mean, each company will have their own system, but at Samsung Biologics, we focus on 3 things, which is program management, risk management, and training. So the program management is about having the right people who have the technical skills to run the high-potent compound program. So it's really about having a very strong EH&S team who can actually provide guidance on safety. And then the roles and responsibilities of all departments are well defined so that they know what to do when it comes to safety issues. Also, you have to have access to technical people such as toxicologists and industrial hygienists who can provide safety consultations. Now, the risk assessment or risk management is about having a good process of identifying what are the requirements for containment, and continuously managing the performance of the containment system is really important. Finally, we focused on the safety training to have a really transparent communication regarding hazard information. And and that will lead to detailed SOPs and procedures for handling spills and gownings in emergency situations as well. So, I want to talk a little bit about the roles of CDMO in ADC manufacturing. We talked about the safety, but there are other aspects of CDMO in ADC. And this is, I think, where the Samsung Biologics has a huge advantage. So as a client, when you choose a CDMO, right, you want a CDMO to have a good understanding of antibody manufacturing and have a manufacturing capability near the ADC manufacturing site is a huge plus. You also want to have a CDMO with an excellent track record of analytical development and testing services. And finally, since there's a lot of moving parts in ADC, the CDMO should be able to manage logistics of supplies, raw materials, intermediates, and finished products. So I want to wrap this presentation to highlight some of the new ADC facilities at Samsung Biologics. We have a very dedicated ADC buildings with dedicated labs and utilities and clean rooms for ADC. We have single-use equipment, but if we need to bring in glass-lined or stainless steel conjugation vessel, that is also possible. All the safety features are based on ISP guidelines, and we're proud to have very safe ADC manufacturing sites. Our target OEL level is 5 nanogram per kilometer, which is one of the lowest in industry, and all our manufacturings are in Class C clean rooms.
So ADCs have certainly demonstrated great clinical efficacy, and moreover, their clinical efficacy is much better than naked antibodies. And when treating patients with an ADC, it does spare them from the systemic toxicity that often limits the dose on untargeted small molecule cytotoxins. Recent improvements in ADCs for difficult to treat therapeutic indications, improved conjugation technology, and a better overall understanding of tumor biology has certainly invigorated the area of clinical interest with respect to ADCs. Manufacturing these highly potent biopharmaceuticals presents a series of unique engineering and chemistry challenges, and as a result, Samsung Biologics has constructed a new facility for this promising class of therapeutic drugs. By leveraging their experience in manufacturing of drug substance and ultimately drug product for clinical and commercial use. Thank you very much for your time.
So Durian Park, Thomas Rohr, both of Samsung Biologics, thank you both so much for taking part in this StatBrand Studio event. For more information, people can certainly visit samsungbiologics.com. Thank you again to everyone in the audience for attending today. Please keep a lookout for a follow-up email with the event recording, and I encourage everyone to share the event landing page across your networks. I also want to remind you that registration is open for STAT's flagship summit in October. More information is available at statnews.com/events. I'm Jesse McQuarters, editor of StatBrand Studio, and thank you for joining today.